Welcome to the 7th Environmental Microbiology lecture. It's lovely to see so many people here. I think we've got a record number of attendees this year. Our speaker tonight is Professor Jim Prosser, who is Professor in Environmental Microbiology in the Institute of Biological and Environmental Sciences at the University of Aberdeen. His research focuses on the diversity and ecosystem function of microbial communities and on the use of molecular techniques to characterize natural communities of microenvironments in soil and aquatic environments. This research has uncovered novel microbial groups involved in biogeochemical cycling processes, in particular nitrification, which plays a central role in the global nitrogen cycle. His achievements have been recognized by election to the fellowships of the American Academy of Microbiology, the Royal Society of Edinburgh, and the Society of Biology, and the presentation of the Francis Clark Distinguished Lecture awarded by the Soil Science Society of America, the Russell Lecture awarded by the Rothamsted Experimental Station, and Distinguished Scientist Lectures at the Lawrence Berkeley National Laboratory, UC Berkeley, UC Davis, and the Craig Bentner Institute. He was awarded a Leverhulme Trust Fellowship to carry out research at the Universities of California, Berkeley, Vienna, and Oxford, and in 2013 was awarded an OBE. So Jim obviously has a very distinguished record, and I'd like to now invite him to give the 7th EMI lecture, which is entitled Unimaginable, Unprecedented Microbial Diversity. Whence, so what, and can we learn from nitrifiers? Jim.
Okay, well, many thanks for that very nice introduction. And obviously, I must begin by saying how delighted I am to be invited to give this lecture. I'm delighted, honoured and flattered to be here. So thank you very much for the organisers, for the Society and for the journal inviting me to give this talk. That's the title. It's a bit, now I look at it after 2 or 3 months after sending it in, I realise it's very pretentious, it's overlong, and embarrassing really, but nevertheless it does hopefully reflect what I want to talk about in this lecture, which is the vast increase in the study of microbial diversity over the last 20 or 25 years, and what I think are the major questions that we should be asking in microbial diversity, and what we might be able to learn from the group of organisms that I have spent most of my life working with, the nitrifiers. Now, it might be good to start thinking about how we measure and assess diversity in microorganisms. And this is the tree of life that derived from work by Carl Woese in the 1980s. And what he did was to revolutionize microbial phylogeny by, rather than depending on or relying on the phenotypic characteristics of microorganisms for classification, he studied the sequences of a single gene, 16S ribosomal RNA gene, in organisms that were then in culture. And this gene has major advantages for phylogeny. It's in the ribosomal RNA gene is involved in encoding for production of ribosomes and which then produce proteins. And that gene is present in every living cell. The 16S ribosomal RNA gene is in bacteria and archaea, 18S gene in eukaryotes. And it's of sufficient length to give sequence length, to give enough variability, to provide discrimination between different groups. And this is and by comparing sequences, and similarities and differences between sequences in different organisms, you can this is based on the idea that can give you information on their relatedness. And this is what came about, this tree of life. And it was a major finding at the time coming from this tree was that bacteria and archaea were as different from each other as the eukaryotes. But that is obviously important, but one reason for me showing this is that if I were to talk to the average person in the street about species diversity and loss of species and so on, they'd probably think about birds, or a bird going extinct, or a polar bear, or whatever. Well, the sort of diversity they're thinking about is here. Here we have animals, here we have plants. The rest of this diversity is microbial. Okay, so you have the microbial eukaryotes here, the fungi, protozoa, and so on. And then here we have the bacteria and the archaea, which previously, as I say, were lumped into a single group. So that's one thing, a major thing that came out of this study. In fact, so instead of having two domains, the eukaryotes and the prokaryotes, we have three. Interestingly, there is now a view that we should go back to the two-domain idea, but with the two domains being bacteria and the archaea, and the eukaryotes evolving from the archaea. But that's another lecture that I'm not qualified to give. So that's all of this was done with organisms that were in culture in the mid-1980s. How do we measure diversity in natural environments? Well, then, and in fact until the early 1990s, the only way we could characterize microbial communities was to take environmental samples and then try and grow the organisms that were present in that environmental sample in the laboratory, and then use either these techniques or the molecular techniques that led to this tree, or the traditional phenotypic characterization of these isolates. And that was fine, except we were always worried. When we did this, we were always worried. We were worried in part that we were selecting for organisms. Whatever growth medium, whatever growth conditions we used, we would select for particular types of organisms that preferred those growth conditions. And therefore, we were always worried that we were not investigating and not characterizing representative organisms and representative communities within the environment. And the other worry was that there were some things that couldn't actually grow, that there were unculturable organisms in the environment that we weren't seeing in the lab. So we were always worried about that. Nevertheless, what we got with these techniques was a view that the microbial community was very diverse. Even using these cultivation-based techniques, they told us that the communities were diverse. One thing I'll mention as well, while I have this slide, is that I mentioned the phrase species diversity a minute ago. Well, it's fine talking about species diversity of animals and plants, and in fact, to an extent for the majority of eukaryotes, because there's a well-established, if not always obeyed, definition of a species, and that is that this comes from the idea of the biological species concept. Members of the same species can interbreed, and those of different species can't interbreed. Well, that's fine for organisms that reproduce by sexual reproduction, but these organisms, the bacteria and the archaea, don't. And there is no reliable, consistent definition of species in the bacteria and the archaea. And that's, well, I say it's a problem. I see it actually as a challenge. It certainly means that when people try to compare the number of bacterial and archaeal species with the number of animal or plant species, I suspect what they're really doing is trying to compare very different things, oranges and apples or pears, or whatever the phrase is. But it's also challenging because it influences the way we study microbial ecology. The fact that we don't have a consistent definition of species means we have to think differently when we study the ecology of microorganisms. Okay, but the ways in which we studied microbial diversity and microbial communities in up to the 1990s were limited. They were based on cultivation. What happened in the 1990s was that people started to develop techniques to enable us to get sequence data from environmental DNA, and to use that sequence data in the same way that it had been used by Carl Woese to classify and identify organisms in the environment without cultivation. And this has now become the standard method for characterizing microbial communities. And it's illustrated in this slide here, where if we start off with what looks a bit like a sample of soil, what we do is add things to the soil to break open, to lyse all of the, well, hopefully, we hope all of the microbial cells in the soil, and then to extract DNA from the soil. and then to purify that DNA, and so that you get a slightly less brown, less contaminated solution, and then to amplify genes using PCR or amplify gene transcript using RT-PCR. Now, typically people will use the 16S ribosomal RNA gene to do this, but you can also use functional genes, genes that encode a particular ecosystem function. And for ammonia oxidizers, that ecosystem function is ammonia oxidation, and I'll be talking quite a lot about this gene here, the AMOA gene, which is the key functional gene for ammonia oxidizing organisms. What you then get is a population, rather than a population of microorganisms, which you have there, you get a population of gene fragments, population of bits of DNA, all the same length, but with very differing sequences. And you assume that the diversity of that population of gene fragments is equivalent to the population of organisms that the diversity of the population of organisms that you have in your soil. And there are various ways of analyzing the sequences in those gene fragments. You can do sequencing, which when we first started doing this involved cloning and sequencing. Then it was a laborious process, and using those sequences either to identify the organism or organisms, or to do phylogenetic analysis, or both. Or then, because sequencing was so expensive, we also used various fingerprinting techniques. In particular, we used this one, DGGE, but you've got death by acronyms there. But we use DGGE, which is denaturing gradient gel electrophoresis. I probably couldn't tell you what the other ones stand for. Okay, so we started doing this. And when people started doing this, they were amazed, because every sequence that we got using this approach, every sequence was different. So I'll talk about this more when we come to the ammonia oxidizers, the nitrifiers. But what we got was incredible, unprecedented, unimaginable, amazing, all those phrases. I just had two of those phrases in the title. We had this massive diversity, much more than we expected on using the traditional cultivation-based techniques. And also we discovered completely new groups. So we had massive, much more diversity than we expected within groups that we thought we knew a lot about, groups that we'd studied extensively in the laboratory. But we also found sequences that formed large, high-level taxonomic groups that had no cultivated representative, that had never been seen in the lab. Now, as you can imagine, that created a lot of activity, and that activity is continuing to this day. So this is a major area of research now in microbial ecology, studying microbial diversity. And a lot of that work was survey work. It was just going out there and looking and seeing what could be found. But for me, it raised two fundamental questions. And one of the fundamental questions was, where does all this come from? Where does all this diversity come from? And the second question was, so what? Okay, you've got all this diversity in say, the soil. Does it matter? Do you need it? Could you lose a lot of it? Could you lose all these, if we call them species, or lose some of these taxonomic groups? Would the soil still function in the same way? Or where's obviously, if you keep taking them away, sooner or later you'll disrupt the soil. But how much of this diversity is important? And also, now we can measure community structure, community composition, without having to grow the organisms. What's that community? Does community composition matter, or is there just lots of functional redundancy in microbial communities? One thing that became apparent was very soon, phrases like amazing, unprecedented, unimaginable, blah, blah, blah. These phrases became redundant. No longer, it was commonplace to find this high diversity. Now I would be, if you told me you had a soil out there that only had 10 different sequence types in it, I wouldn't believe you. Okay, so it's no longer amazing and unprecedented and unimaginable and so on to get this high diversity. It's what we expect. That doesn't stop people using these phrases in papers they write and talks they give to try and get the papers in better journals and so on. But nevertheless, it's commonplace now. But the questions are still far, those two major questions are still far from being answered. Okay? And what I want to do today is to talk about how we've tried to address some of those ecological questions as applied to the nitrifiers, the ammonia oxidizers, and how what we've done may be illustrates some of the pitfalls that can be that you can experience studying microbial diversity, and maybe some of the benefits of the approaches that we've used. And before I do that, I just want some sympathy for anybody who doesn't work on soil microbial ecology. Okay, so what we're dealing with is this. This is a typical. This is the sort of scale that we sample at. We typically sample 1 gram of soil and see what's in there. We might measure substrates or products or communities in that sort of scale. Now that gram of soil will contain about 7 billion bacteria. So there's the planet, and that's got about 7 billion people. Okay, so a bacterium sees a gram of soil in the same way that we see the planet in terms of human population. Okay, so there's lots of them, and most of them won't meet each other in the same way that we won't meet most of the 7 billion people on the planet, despite how many downloads we get from this lecture. And that in itself is a problem. When we sample our gram of soil, we're averaging everything. We get a solution of DNA from a gram of soil and analyze the community in that gram of soil. And this is what soil looks like. This is X-ray computed tomography of round about a gram of soil. And it's designed to show the pore structure and the complexity of the pore structure in soil. So you can see how complex this is. If you've got an organism here and you're wondering about how it's going to interact with an organism over there, it's probably never going to see it. Except that it's never going to swim all that way. For an organism, that's like you trying to swim to India from here. Okay, that's not going to happen. But you can get a plane, and if the equivalent of a plane, maybe not quite the equivalent, might be a worm crawling through this soil. That will transport organisms across the soil and maybe enable them to interact. And they'll be interacting inside the worm as well. So I'm looking for sympathy. But you could also say, well, it's your own fault. We go into the soil knowing it is the most complex environment in the world. So really only very brave or very stupid people become soil microbial ecologists. And you can decide which I am. Okay, so what I want to talk about is nitrifiers and nitrification. So a quick run through what nitrification is. So nitrification involves 2 groups of microorganisms, ammonia oxidizers and nitrite oxidizers. And there are typical ammonia oxidizer and nitrite oxidizer cells. I'll be focusing mainly on ammonia oxidizers, and the ammonia oxidizer that we have there is Nitrous ammonius europaea, which is the lab rat of ammonia oxidizers. So it's the ammonia oxidizer about which we know most in terms of its physiology, biochemistry, genetics, and so on and so forth. So we know that's our lab rat, that's our E. coli, if you like. Now, ammonia appears in the soil from 3 main sources, from animal excreta, from decomposing organic matter, and from fertilizer nitrogen. Nowadays, most of it comes from fertilizer nitrogen. There's 115 million tons of nitrogen fertilizer added to the soil annually, and costing tens of billions of pounds, more than is produced naturally. The ammonia goes into an ammonia oxidizer cell, it's converted to nitrite, the nitrite goes out of the cell and then into a nitrite oxidizer, and the nitrite is converted there into nitrate and then leaves the cell. And then the problems start, okay? Because whereas ammonium will stay in the soil just waiting for a plant to use it and grow, nitrite doesn't. It can be denitrified, and one of the products of that can be nitrous oxide, which is a greenhouse gas, and obviously that leads to fertilizer loss because it disappears as a gas. Or the nitrate can leach through the soil into groundwater, where it pollutes the groundwater. And if the levels get high enough, you're not allowed to drink it. What you have to do is dilute it so it's just below the level, and then you can drink it. So you have environmental problems, groundwater pollution and greenhouse gas production, and enormous economic losses through fertilizer loss. So in some intensive agriculture regions, less than 30%, up to only 30% of the ammonium-based fertilizers end up in the plant. 70% of it can be washed away. So if I said tens of billions of pounds are spent on fertilizers annually, 70% of that is lost in many places. So Someone once described, Elder Paul, an American soil scientist, described nitrification as the biggest disease in agriculture, and this is why. Okay, so these organisms are autotrophic. They get the carbon by fixing carbon dioxide, and what you see there is what you'll find in textbooks, and this was sort of dogma, but we now know that these organisms are much more diverse physiologically. So they can both groups can take up organic matter, although ammonia oxidizers can't grow on organic carbon, but they can both assimilate it. The ammonia oxidizers can take up urea and convert that into ammonia as an alternative source of ammonia, if you like. And both groups denitrify and produce nitrous and nitric oxides themselves rather than waiting for the denitrifiers to do it. So again, they contribute to atmospheric pollution. And they can also use methane rather than ammonia to produce methanol. Okay, so that's ammonia oxidizers, physiologically more diverse and therefore involved in more ecosystem processes than the average textbook will tell you. But what about so that's by way of introduction. What about the ecological questions that we want to ask? Well, I'll now introduce the ecological question that I will focus on today, and it's what I like to call the paradox of nitrification in acid soils. So if we look at cultivated ammonia oxidizing bacteria and look at the effective pH on their growth rate, this is what we find. None of them can really grow below pH 7. They all have pH optima for growth round about 8. But of these organisms that are plotted there, whose behavior is plotted there, the majority have been isolated from acid soils. So that in culture, in liquid batch culture, they can't grow under acid conditions, but but most of them have been isolated from soils, sometimes with pH values as low as 4.5. Now, the explanation for this is given on this slide. If you imagine an ammonia, here's our nitrous ammonium europaea cell, growing in liquid medium containing ammonium. Now, in liquid, there'll be an equilibrium between ammonium ions and ammonia and hydrogen ions. When the pH, and what happens is the ammonia diffuses into the cell, some of it diffuses out, but while it's in the cell, some of it will be converted to nitrite, which will diffuse out of the cell, some of it will go back, but what's left outside will equilibrate with nitrous acid. Okay. Now, if you reduce the pH, you increase the hydrogen ion concentration, and that pushes that equilibrium to the left. So what happens is the ammonium concentration increases, ammonia goes down, there's less in the cell, so there's less nitrite produced. And what nitrite does get produced, most of it when it gets out of the cell, is there as nitrous acid. So an ammonia oxidizer growing at low pH has two big problems. Hardly any substrate, because the substrate's ammonia rather than ammonium, and a lot of an inhibitor, nitrous acid. So that fits with what we see in liquid culture. But it doesn't fit with what we see in the environment. So if you look at the measure gross nitrification rates and look how they vary with soil pH, so this is gross nitrification rate in soil, and this is a meta-study carried out by Booth and co-workers. What you find is that pH doesn't really have much of an effect on gross nitrification rate. In fact, some of the highest rates are in very acid soils with pH values around about 3.5 or 4. I mean, they drew a line through that. I'm not sure you believe that line, but it's going the way that you wouldn't expect it to. It's suggesting, if anything, that the rates are higher at low pH. So we have an interesting scientific ecological question. We get nitrification going on in these acid soils when the organisms that we believe are doing it, that carrying out the first step in nitrification, ammonia oxidation, they can't grow below pH 7. That's an interesting question. Is it an important one? Well, I would say yes, wouldn't I? But if you look here, you have a map of the Earth with, well, Graham Nicol, my colleague, said this is what would happen if you wrapped a piece of litmus paper around the planet. Okay, because the red bits there are low pH soils, the blue areas are alkaline soils. A lot of the planet is covered in has acid soil. And I think one estimate is about 30% of soils have pH values less than 5.5, and that includes 50% of arable land. So it's an important problem because in these acid soil regions, people will be putting ammonium-based fertilizer onto the soil, it'll be converted into nitrate, it'll be nitrified, and we don't know how that can take place because the organisms that we believe do it can't grow below pH 7. So that's where we were in the early 1990s. We had various ideas about how this might take place, which we'd already been studying for 10 years. For example, the bacteria can grow on surfaces, and that might protect them a bit from low pH, and there are other potential explanations. But what we could never do was find out whether soil pH selected 4 different groups of ammonia oxidizers, whether you had acidophilic ammonia oxidizers or alkalinophilic ammonia oxidizers. Then along came these molecular techniques. And they came along to me I was at a conference on a completely different subject, and a guy called Martin Embley came up to me on the bus on the day out, or the trip out, and asked me if I knew anything about 16S ribosomal RNA gene-based methods, and I didn't. He was a molecular phylogenist, and he suggested that we get together and use this approach to see if we can study nitrification in natural environments. And that was the start of what was an amazingly productive and enjoyable collaboration. Well, it was for me, I'm not sure about Mark. So what we did was to look at sequences of 16S ribosomal RNA genes of a few cultivated ammonia oxidizers that had been sequenced at the time, and use those sequences to design primers that were specific for those ammonia oxidizers, and then use those primers to amplify from soil 16S ribosomal RNA genes just of these bacterial ammonia oxidizers. And this is the result of that study. And what I'm going to do now is plot 2 phylogenetic trees which contain those sequences, the sequences that we got in this first study. I've been showing this slide for nearly 20 years now, and various parts of this phylogeny have been improved on significantly, but the major points that I'll mention and that I'll bring out of this slide stand today. One day I'll get bored showing it, but sorry, today is not the day. It still excites me, this slide. Okay, so what I'm going to be doing is putting on this tree sequences we got from pure cultures, the few pure cultures that were available at the time, soil sequences we got from DNA extracted from soil, sequences from marine environments, and sequences from wastewater treatment processes. And we have 2 genera of ammonia oxidizers. It's a bit more complicated than that, but let's say we have two genera, Nitrosospira and Nitrosomonas. So for Nitrosospira, here you see the pure culture sequences. Then we have some sequences from soil, blue sequences from marine environments, green sequences, more green sequences from soil, and then the same for Nitrosomonas. If I press the button. Okay, the pure culture sequences, marine, sewage, and a mixture of soil and marine sequences. Okay, so first of all, what we found is what I mentioned earlier. Every sequence was different. Okay, no 2 environmental sequences were different. And no environmental sequence was the same as the sequence of a cultivated organism. So much, much more diversity than we expected. And the sequences that we got were not the same as those of organisms we knew about. We also found that we could cluster these sequences. And particularly for Nitrosospira, we found that those clusters were associated with the environment from which we got the sequences. So we have cluster 1, which is marine, cluster 5 is marine, and then cluster 2, 3, and 4 just have soil sequences. You'll also notice that the cluster 1 and the cluster 5 groups don't have any white sequences in, i.e. there were no cultivated representatives of those sequences. We presume those organisms are ammonia oxidizers, or the organisms from which those sequences originated are ammonia oxidizers, but to this day we haven't managed to enrich or isolate representatives of those sequences. And then down here we've got our lab rat. So there's Nitrosomonas europaea. Now, Nitrosomonas europaea was originally isolated from soil, and it was thought to be a typical soil microorganism. typical soil ammonia oxidizer. But if you look where it is, it's in the nitrous amonis group, nitrous amonis genus, whereas most of the soil sequences are nitrous aspirers. So our lab rat is not a typical soil ammonia oxidizer. And for me, Martin Embley always laughs when I give this analogy, although I did check it out with him before the first time I did it. But this for me is analogous to a bird ecologist who's based everything he thinks about bird ecology on the basis of domesticated birds. So he studied canaries and budgies and so on, pigeons maybe, and based everything he knows about bird ecology on the behavior of those organisms. And then he gets a new technique. And the new technique is to go outside and open his eyes, maybe even with binoculars. Imagine how you would feel as that bird ecologist if all of a sudden you saw puffins and penguins and thrushes and all the other birds that you see. Well, that's how we felt, or at least that's how I felt. I'm not sure how Martin felt, but that's how I felt when we got these data. This told us that there was an incredible amount of diversity out there, stuff that we just didn't know about. And that was really exciting. Now, I hesitate to say this because it's being videoed, but for the animal ecologist, for that bird ecologist, it's okay, because you can then carry on doing ecology. You can follow birds around to an extent, and you can see whether they eat berries or worms or roadkill, and you can see where they live and so on. We can't do that. Our ammonia oxidizers are about 1 micron, 2 microns in length. So just having this sequence data didn't tell us what they did, other than we hoped that they could oxidize ammonia. But we couldn't follow the cells around and find out what else they did. So we had to be cleverer. We had to, and that's what happened. People developed techniques, again, techniques which didn't involve cultivation, but techniques to measure or estimate activity. But it's much more difficult. So this is where I get a bit worried. If you ever see an animal ecologist stood next to a microbial ecologist, you'll know who's cleverer, because animal ecology is much easier. We have to it's much more difficult doing microbial ecology. And if you see a microbial ecologist stood next to a plant ecologist, it's even easier doing plant ecology because you don't have to run after things to see what they're doing. You can just go up to them and ask them. So what we then did was to use these techniques, this approach, to look at the effects of soil pH on the ecology and the distribution and the community ecology of these ammonia oxidizers. And we're blessed in Aberdeen for many reasons, but one of the reasons we're blessed in Aberdeen is we have the Gravestone pH control plots. And these are plots of soil which have been kept at pH values ranging from 4.5 to 7.5 since 1961. And if you want to see them, go soon, because they're about to build a ring road through this area. We'll probably lose these plots unless we can find a way of preserving them in some way. But anyway, what we did, John Stephen, postdoc jointly supervised by Martin and myself, sampled soil along these plots and looked at the ammonia oxidizer communities using this fingerprinting technique, DGGE, that I mentioned earlier, to see if there was selection for different ammonia oxidizers with pH. And there was. Okay, so you can see this cluster 2 group decreases in relative abundance as pH increases, cluster 3 increases, cluster 4 maybe peaks in the middle, cluster 6A is not so important here. So this indicated to us that there might be pH selection for bacterial ammonia oxidizers, at least in this soil, and that this cluster 2 group might be important in this respect. And we probably would have, we probably should investigate that in more detail. But after this revolution of the 16S ribosomal RNA revolution, we then were impacted, if you like, by another revolution. And to talk about this revolution, I want to go back to the tree of life that I showed earlier and talk a bit more about archaea. Now, at the time that this tree was drawn, all of the archaea that were known about were extremophiles. They were scary things. They grew in environments that nobody would want to go to. They had very high temperatures, very low pHs, pH 1, pH 0. They can even grow in Coca-Cola. At the very high salt concentrations and so on. So archaea were thought to be extremophiles. But when people started designing primers for these organisms and going and looking in natural environments, they found representatives of what originally was called group 1 archaea everywhere. So they found them in the ocean, they found them in lakes. If you look in soil, I'm not sure, in the middle of London, I'm not sure there is any soil outside, but if there is, 4% of the prokaryotes will be archaea. They grow, they're not all extremophiles. An awful lot of them are. It's probably the majority of them may not be. But there wasn't a cultivated representative of these group 1 archaea, so nobody knew what they did. And if you looked at the phylogeny, as Graham Nicol, my colleague, well, at this time he was a postdoc of mine, but he's now professor of 3 days. So Graham, with Christa Schlieper, constructed this phylogeny of the crenarchaeal 16S ribosomal RNA genes. And there in white you see what we always knew about, the extremophiles. But then they found sequences, or other people had found these 16S ribosomal archaeal sequences in a whole range of other environments. By this time they were called the crenarchaea, and well, that's about to change in the next 2 seconds, because down here in this bottom right hand corner there is this organism here, Nitrosopumilus maritimus. Now Nitrosopumilus maritimus was isolated by Martin Konecker at the University of Seattle, and it is an archaean and it's an ammonia oxidizer. So this sent reverberations throughout the ammonia oxidizer community and other communities as well, I suspect, because this said that after 100 years of studying nitrification and thinking it was done by bacteria, this now says that it's not just bacteria that do it, it's archaea as well. This is a bit like your bird ecologist thinking you knew everything about things that could fly, and then somebody discovering insects. I mean, it's similar in impact. Okay, a completely different phylogenetic group with different properties can do ammonia oxidation. And in fact, we now know that all of these, this whole part of the phylogenetic tree contains everything in there that we know about, either can oxidize ammonia in culture or contains, if it's not culturable, contains these AMOA genes, contains the genes that you require for ammonia oxidation. Doesn't mean everything there can oxidize ammonia, but everything we know about does. So it's now given a new name, the tomarchaea. Okay, so we discovered this, we found out about this, and that then created frenzied activity similar to that had happened in the early 1990s when people first got the 16S techniques. But now what people did was to go around the world sequencing or looking for sequences of archaeal AMOA genes. And they found they were everywhere. Any environment you care to look at, you'll find these sequences. And in some soils, well, in many soils, these archaeal AMOA gene sequences outnumber those of bacteria. And that suggested that possibly, but it's only possibly, the archaea may be more important for soil nitrification than bacterial ammonia oxidizers. And because these belong to not just two different phylogenetic groups, but two different domains, major domains, the different properties associated with those, distinguishing those domains, may mean that they have different, very different ecology, and that they may behave differently in terms of converting our expensive fertilizers into atmospheric pollutants and groundwater pollutants. But we were just interested in pH at this time. Well, I'm only talking about pH now. So what we did was to go back to these Cravestone soil plots. This is 10 years later, and it's quite a brave thing to do, to ask one of the people working for you to go back and repeat what was done 10 years earlier by somebody else, because who knows? If they want to give you good data, they may just give you good data, whether it's real or not. But anyway, Graham, was then a postdoc, went back to these Cravestone plots and did the same as John. He used DGGE to look at the communities in these different pH plots and got pretty much exactly the same results as John. So some sequence types, some phylogenetic groups were dominant at neutral to alkaline soil pHs, and some were selected by acid soils. And the groups were the same, the acidophilic and the neutrophilic or alkalinophilic groups were the same. But they also looked at so that's just saying it confirms the results that John got, and says also that it's a very stable selection because it's the same as it was 10 years ago. And then he looked also at these AMOA genes from the archaea and found the same. Some were selected at neutral to alkaline pH and some at acid pH. But he also did another thing. To try and assess activity rather than just presence. The presence of a gene tells you an ammonia oxidizer organism is there, but it doesn't tell you whether it's active or not. But if it's active, if you add ammonium, then it's likely to switch on the AMOA genes. So what he did was to count AMOA genes and to count AMOA gene transcripts and calculate the ratio of transcripts to genes as a measure of activity. And what he found was that activity fell significantly as the pH increased for the archaea, but it increased a bit, not quite so much, but it increased with pH for the bacteria. And that suggested that the archaea may be much more important in these acid soils. Now we told this story, we suggested that archaeal ammonia oxidizers might be important in acid soils. And people looked at the data and I'm not joking, they said, oh, that's just a strange Scottish soil. It's a local effect. It's not really. Now, I mean, I can understand to an extent what they're saying, but what this inspired us to do, or goaded us into doing, was a global study. And the way we did a global study was work done by a postdoc in the lab then, Cécile Goubry-Ranjan. And what she did was to go into the gene sequence databases and look and pull out every archaeal AMOA gene sequence for which there was information on soil pH. And she did cluster analysis of these sequences and got 18 clusters that you can see there, and then looked at the frequency of occurrence of each of these clusters in soils of different pH. And so what she got was this. So for each of these clusters, you can see whether they are, for example, cluster 14 here, whether they dominate, whether they're only found, in this case, in acid soils, or, as in this case, only found in alkaline soils or in a mixture. And she classified each cluster as acidophilic, which is given by the red dots, moderately acidophilic, given by the pink dots, or alkalinophilic, indicated by the blue dots. And we then used that information to predict what we would find in 47 UK soils that were chosen to cover a wide range of pH. Okay, so here we did high throughput sequencing of AMOA genes in these soils, which we got from CEH. In fact, we didn't even get the soils from CEH. It was even better. We got the DNA. So we didn't even have to extract the DNA. And what we got agreed pretty much with what we found globally. So the globally defined, if you like, acidophilic groups were also dominating acid soils in the UK. So the regional and global distribution of these organisms, different groups, agreed. You'll see a couple of times, a couple of cases there when they don't quite agree, and that's usually where there's very few sequences or very few soil samples. And we also did the same for our gravestone plots. And what she found was that of the six most abundant clusters there, I think it's only 5 on this slide, we have one alkalinophilic group, 3 moderately acidophilic groups, and 1 very acidophilic group. Okay, and that is really acidophilic. I mean, the red there, you can see most of those are in soils pH less than 4.5. Now, then one of these amazing coincidences happened, amazingly good coincidences happened, because elsewhere in the lab, a PhD student, Laura Leiteverta, was trying to enrich ammonia oxidizers from soil, archaeal ammonia oxidizers from soil. And she got this organism here. There's a couple of pictures of it. Oops, I've gone too far. No. Yes, I have. Right. Go back. Okay, so is it going to go back? Yes. Okay, we call this organism Nitrosatalia divanatera. And again, it's an autotrophic archaeal ammonia oxidizer. It's called Nitrosatalia divanatera. Maybe we should explain that. Nitroso means ammonia oxidizing. Talia means rod. Terra is Latin for soil. And Divana is an old Roman name for Aberdeen. So it's an ammonia oxidizing rod from an Aberdeen soil. Sounds a lot better in Latin. But why am I bothering to tell you about this? Well, if we go back to the slide I showed you earlier, showing the effect of pH on the specific growth rate of ammonia oxidizers, that's the bacterial ammonia oxidizers there, some of which we obtain from acid soils, but none can grow below pH 7. There's Nitrous Italia devanatera, acidophilic. It grows at pH 4, pH 5, pH 5.5. Won't grow at 6, and it won't grow at 7. So it doesn't grow where the bacterial ammonia oxidizers grow. It doesn't grow at their pHs, but it grows under these acidophilic, under these acid conditions. And when we do the phylogeny, 16S phylogeny for the Tomarchaea, this is where Davana strain or acidophilic strain falls. There it is in the tree. And it was called group 1.1A associated. It's now called the Nitrous Italia group. It's named after our strain. And the best bit, if we bring in the phylogenetic analysis that Cecile did, there's our acidophilic group that's globally distributed, and Nitrous Italia is a member of that group. So this odd little finding that we had in this strange soil in Scotland has global significance. This organism that we isolated, the first acidophilic ammonia oxidizer in the world, is an archaeon, and it belongs to this group here, this phylogenetic group, which is distributed throughout the world in acid soils. Now, that's all very well. But do we have another laboratory? Is this like Nitrous ammonius europaea? Is it a lab rat? Well, what Graham did was, by this time he was supervising. He wasn't just a postdoc, he was a lecturer supervising. Nevertheless, he went back into the lab and did the microcosm experiment to find out if this organism could really grow under these conditions, under these acid conditions in the soil. So what he did was to set up soil microcosms, basically tubes with some soil in, and he measured nitrate concentration with time to see if there was nitrification occurring. He measured the abundance of this Nitrous Italia strain. So this is a specific assay for Nitrous Italia devanatera, and you can see that there's some there at day 0. And then he used DGGE to look at the community, how the community changed. And there's the Nitrous Italia band in yellow, circled in yellow on our DGGE gel. Again, there's some there at day 0. In some of the microcosms, he added acetylene, which is a potent inhibitor of ammonia oxidation. When he did that, he got no nitrate production. He sort of killed off the Nitrous Italia, which is a bit unusual, actually. Normally, the ammonia oxidizers survive acetylene, but this one didn't. Oops, it's going too fast. And you can see the band decreasing in intensity in the gel. But when he didn't have the acetylene there, we got plenty of nitrate produced. We got an increase in abundance using our qPCR assay, and we also saw an increase in the intensity of the band in the gel. And he added 12C carbon dioxide, or natural atmospheric carbon dioxide, to some of the tubes, to some of the microcosms, but to others he added 13C labeled carbon dioxide. Now, it made no difference to these data here, but the reason he did that was to use one of these techniques that have been developed to look at the activity of microorganisms in soil, specific microorganisms in soil. And this technique is called stable isotope probing. And what we do here is we incubate our soil in the presence, in an atmosphere, of either 12C carbon dioxide or 13C labeled carbon dioxide, heavy carbon dioxide. Now, any organism growing in the soil and fixing carbon dioxide, which the ammonia oxidizers do, will assimilate either 12 or 13C carbon dioxide. If they assimilate 13C carbon dioxide, all of the carbon, the organic carbon in their cells, will be 13C labeled and will be heavier. So if we extract DNA from the soil and then carry out ultracentrifugation, what you will get when you spin the DNA down is a gradient going from 12C DNA down to 13C DNA. And then if you do molecular analysis on fractions from this gradient, the organisms down here, the genes that you get down here and you quantify down here, the AMOA genes there, will be from organisms that have assimilated the carbon dioxide, the 13C labeled carbon dioxide. And any AMOA genes here will be from organisms that haven't assimilated carbon dioxide. So this is a way of finding out which organisms use specific carbon compounds, in our case carbon dioxide, during growth. So what did he find? Well, this is what he found. Okay, so if we looked at the proportion or the relative abundance of MOA genes as a function of the mass of the DNA, so here we're going from light DNA down to DNA, we're going down this gradient. If he incubated with 12C carbon dioxide, he got a peak here with a buoyant density of around about 1.7. When he incubated the microcosms with 13C labeled carbon dioxide, this is what he got, a complete shift in the peak, because these are organisms that have been growing in the 13C labeled incubated microcosms, and that's where the MOA genes were, the archaeal MOA genes of Nitrous Italia Terra. So this whole experiment shows that the archaea are important in oxidizing ammonia in this soil. It's being carried out by Nitrous Italia, and it's growing autotrophically while it's doing it. So this is very strong evidence for this organism being responsible for acidophilic nitrification in this soil. Now, what we could have done was then say, fine, well, that's job done. Okay, we've solved the paradox of nitrification in acid soils, and we may have done. To an extent, we probably have. But we must remember that what we found out before molecular techniques need not necessarily be wrong. So the data that I showed you earlier, which indicated the bacteria may be some of the bacteria may be selected at low pH, we maybe should have a look at that. But all the evidence we have so far suggests that the archaea are much more important than bacteria in these very acid soils. Okay, now where do we go from here? Well, one of the way Cecile has gone, Cecile Gubri-Ranjan, she's now on an independent fellowship in Aberdeen, and she's gone the way of evolution. So what she's doing is using these same data, and also new data that we're getting, to study the evolution of these organisms. So she's addressing, and hopefully answering, the where does this diversity come from experiment. Why do we have such high diversity, and how has it developed over the years? So here's this is a tree based on the same data I've been talking about, where you can see the origin of each sequence is indicated by the color at the end of the tree there. So these are mainly from acid soils, these are mainly from alkaline soils. And this shows the development, the phenotypic development that you can estimate using these sequence data. And then, so there's all the clusters, and that's showing how the phenotype, this pH phenotype, has developed in ammonia oxidizers, how they've developed and adapted to different pHs. And by looking at the MOA gene sequences, she can determine diversification rate. Okay, so this shows whereabouts in the tree diversification has been faster, where it's been slower. And along the bottom here, you have relative time. We can estimate real time, but only very roughly, because we don't have fossils to base this on. But this is probably between 400 and 700 million years. I mean, it's quite a range there. But nevertheless, that's the sort of timescale we're talking about there. And from those data, you can calculate what animal and plant ecologists would call speciation rate. So here, she's done that based on the MOA gene sequences. Here's the diversification rate for these ammonia oxidizers. Now, the interesting thing here is there's obviously been a burst here in activity, in evolution. So big increase in speciation rate over this period here, and then it levels off. Now, this doesn't mean it levels off at a certain number of species. It means the speciation rate levels off. So there's still a lot of speciation going on up to the current day. Now, what happens normally when you do this with animal and plant communities is that when an organism invades a new environment, then you get a lot of adaptation to fill the new niches. But once the niches are filled, the speciation rate starts to decrease. So what we'd expect is maybe this increase, but then a decrease once we've filled the niches. But that's not happened with these organisms, not happened yet, and there may be reasons why it may never happen We don't know. So we don't really know why this is happening. It may be something to do with the complexity of the soil or the fact that we don't have real species in prokaryotes. We don't really know, but it's a phenomenon that we want to investigate, or she wants to investigate further. And if you do the same for phenotypic adaptation, then you get a very similar curve. And that indicates that the diversification that we see, the high diversity that we see, is due to speciation resulting from adaptation to pH. And there's another slide here which indicates that these circles on the trees here with this analysis indicates points where there's been shifts in rate, increases in rapid periods of rapid diversification, and you get the same, the circles are in the same place whether you look at diversification or adaptation to pH. So that's one way, that's one thing we're doing, looking at where does this diversity come from, why is it so big? Laura has been doing, she's become a biochemist, and we've characterized the genome for this acidophilic strain, and these are genes here which indicate common mechanisms for adaptation by organisms for moderate acid conditions. So these are genes and pathways that are involved in maintaining cytoplasmic pH when you change the external pH down to maybe 4 or 5. But what's interesting is when she looked at ammonium transport, she found that if you did phylogenetic analysis of ammonium transport genes in bacteria, that's the bacterial ammonia oxidized. Well, if you did a phylogenetic analysis of genes in bacteria and archaea, the bacterial ammonia oxidizers, their ammonia transport systems fell in this region here, and they transport ammonia, whereas the archaea have ammonium transport systems which are in this part of the tree, and they transport ammonium. So this may be, what this suggests is that if you have mechanisms for maintaining cytoplasmic pH around about neutral when the external environment is moderately acidophilic, and you can take up ammonium rather than ammonia, then you're able to grow at these low pH values. Because remember, as the pH decreases, ammonia, the equilibrium moves from ammonia across to ammonium. So at pH 4.5, there's hardly any ammonia there. It's nearly all ammonium. And we've been answering other questions. The pH question is the one I've focused on today. One of the horrible things to do in a lecture like this is to not include everything you want to, because there's lots of other projects and lots of other slides I'd like to show you, which I've not been able to. But one of the things we've started looking at is drought resistance, whether the extent to which ammonia oxidizers can survive periods of drought, and then how fast they recover when it rains, and whether archaeal and bacterial ammonia oxidizers are as resistant and resilient as each other. And the basis for this is that, or the basis for suspecting there may be a distinction, is that there's some evidence that the archaeal ammonia oxidizers have higher substrate affinity. They can grow at reasonably fast growth rates at very low, much lower ammonia concentrations than bacterial ammonia oxidizers, but also that they're more sensitive to high ammonia concentrations. High ammonium concentrations inhibit ammonia oxidizers. And it seems that, very limited evidence at the moment, but it seems that the archaea may be more sensitive than the bacteria. Now, if you have a soil with ammonium in, and it dries, then what happens is you end up with just a thin film of water on the particulate material, and the ammonia will be concentrated there. So if the archaeal ammonia oxidizers are more sensitive, then you'd expect them to be less resistant, and that's what we found, less resistant than the bacteria. Then when it rains, what you get when it rains is a flush of activity, because the mineralizing organisms in the soil start degrading the organic nitrogen straight away, so you get a big increase in ammonium concentration. So again, the ammonia oxidizing archaea suffer, and again, that's what we've found, that when the bacterial ammonia oxidizers recovered much better than the archaea. Okay, there's other questions we're looking at which I don't have time to go into. But what I'd like to do is to sum up by reflecting on what we've done over the last 20 odd years with studying the ecology of ammonia oxidizers and how the lessons we have learned may apply more broadly in microbial ecology. Some of the things we are still grappling with. The fact that we don't have species definitions is a problem when we're studying these organisms, and whether there are strong links between phylogeny and function. These are still open questions. But one thing that I would say we have learned is that it is, well, you don't get answers unless you ask questions. Okay? And virtually all of the work we've done has been attempting to address questions or to test hypotheses. We have been involved in some of the more descriptive survey type work or correlation type work where people just go out looking for relationships between communities and soil characteristics. I went into some of these studies innocently and have been dragged kicking and screaming into some of them more recently, but I've never found that approach to be very productive. Whereas where we've attempted to address specific questions and had ideas and potential explanations before we've done the experiments, and where we've adopted an experimental approach rather than just looking and seeing and just seeing what's there, that has been much more productive for us. I suspect most talks on microbial diversity these days, when they look forward, they start talking about metagenomics and metatranscriptomics and metaproteomics and so on, and also the amazing techniques that are available that the physicists are developing for us to image soil. They are incredible techniques. But unless those techniques are addressing important questions and can address important questions, then they will be useless. And for me, that's probably the most important thing I've learned, and not just from this work. It's important things I was taught when I did my degree and when I did my PhD. That if you don't ask questions, then you don't get answers. And I think that is the biggest message I can give for what we've learned in ammonia oxidizer community ecology that we can put into the rest of microbial community ecology, which isn't to say that we're the only people asking questions. Lots of people are asking questions, but probably more people are just going out and looking. And I think the field will advance much faster if we ask questions rather than just going out and looking. So if I have a message, that's it. So thank you very much.